Hello everyone, welcome back to our next class non-coordinates unit 1 Protista, Parazoa and Metazoa. Today I will be discuss about life cycle and pathogenicity of Entamoeba histolytica. As you know that Entamoeba histolytica is a protozoan parasite. It is intestinal parasite and it causes the amoebiasis disease in human being. Let's know about Entamoeba histolytica life cycle. Entamoeba histolytica are pathogenic amoeba that are widely known for causing intestinal and extra-intestinal infections in human beings. Entamoeba histolytica falls under the phylum Protozoa, the class Sarcodina, and the order Lobosa. Entamoeba histolytica causes dysentery by invading the mucosa and submucosa layers of the large intestine in human beings. Entamoeba histolytica are commonly found in tropical and subtropical countries. The life cycle of Entamoeba histolytica begins and ends inside one single host, that is an individual human being. So it is very interesting because it passes its whole life cycle within the one host, that is man. Here in the figure you see Entamoeba histolytica life cycle. So this is cyst, the measuring about 10 to 12 micrometer, and this measure cyst when ingested by man through eatable things or through contaminated food, it swallow and then it reaches the RBC. Within the RBC, it forms trophozoite. This trophozoite is measuring about 12 to 20 micrometer. And here see the chromatin also shown. And this is pseudopod, which is the characters of amoeba. Now it forms colonies. The mucosa means cyst formation takes place. This cyst passes in the stool in the human body. No intermediate host. So the flies directly carries from stool and this cyst again get ready to infect the another person. So this cycle is repeated. Now you see the stepwise how it infect the human body. Number 1, cyst, stage 1. Infection occurs by ingestion of mature cyst through facially contaminated water or food, as I mentioned above. Due to protection from walls, cysts survive several days and sometimes weeks. They are responsible for transmission. Up to this stage, they remain in wall. Now, second excision, that is the stage 2. Excision is the process by which cyst transforms into trophozoites. When the cyst enter the ileum of the small intestine of the host, the process of excitation begins. Trophozoites are released in the small intestine, and from here they migrate to the large intestine. Now they follow the step 3, trophozoite, stage 3. Trophozoites are unicellular parasites that measures from 14 to 18 millimeter in diameter. They multiply in the small intestine by binary fission to produce cysts that exist via human stool. Several trophozoites remain inside the lumen of the small intestine. The rest attach themselves to the intestinal mucosa, enter the bloodstream, and further grow in the extraintestinal region of the host like lung, liver, brain. So you imagine it not only the intestinal, it shows the extra-intestinal because it migrates to the other part like liver, lung, brain also. Here, the schematic figure of life cycle you see the trophozoites which passes in stool. They shows the pre-cyst stage which is rich in the glycogen. This pre-cyst form the cyst through hyaline cyst wall. Then hyaline cyst wall it forms the 10 to it is measuring about 10 to 20 micrometers and it is 2 to 4 nuclei form. Here no glycogen, no chromatidal bodies which is present in this pre-cyst stage. Now this cyst, if it is the semi-formed stool, it formed a pre-cyst or cyst and it directly infect human. If it is formed stool metacyst, then these metacyst trophozoites like mature trophozoites again it infect the human. So it's two way it can be infect. So when in enter the human, then it reaches to the in small intestine, then existing takes place, then metacystic trophozoites form, and then it is trophozoites. So in such a way, the cycle is repeated. So another figure you see, so here shown the human body, the mature cyst ingested somehow, and then here rest part takes place, means cyst formation takes place, the cyst trophozoite form, and again this cyst or trophozoite passes through stool, and then again it's ready to reach through contaminated food to human body. So this is the mechanism of the life cycle of Entamoeba histolytica. Entamoeba histolytica is an anaerobic parasitic amoebozoan, part of the genus Entamoeba, predominantly infecting humans and other primates causing amoebosis. So you imagine it not only human, some other primates also it's get infected. Entamoeba histolytica is estimated to infect about 35 to 50 million people worldwide. So up to this much figure, 35 to 50 million people it's get infected, Entamoeba histolytica infection is estimated to kill more than 55,000 people each year. What is strange? So 55,000 people only die due to the Entamoeba histolytica infection. So can you imagine? Mammals such as dogs and cats can become infected transiently but are not thought to contribute significantly to transmission. The word histolysis literally means disintegration and dissolutions of organic tissues. This is literally meaning. Diagnosis is confirmed by microscopic examination for trophozoites or cysts in fresh or suitably preserved vessel specimens, smears of aspirates or scrapings obtained by proctoscopy, and aspirates of asbestos or other tissue specimen. A blood test is also available but is only recommended when a healthcare provider believes the infection may have spread beyond the intestine gut to some other organ of the body, such as the liver. In that case, it is recommended. However, this blood test may not be helpful in diagnosing current illness because the test can be positive if the patient has had amoebiasis in the past, even if they are not infected at present. Stool antigen detection and PCR are available for diagnosis and are more sensitive and specific than microscopy. So generally this kind of examination is done for detection of the Entamoeba histolytica, cysts or trophozoites. Now you see the pathogenesis. Entamoeba histolytica causes tissue destruction which leads to clinical disease. Entamoeba histolytica includes tissue damage by three main events: direct host cells, death, inflammation, and parasite invasion. So when it attacks the cell, means host cells, the host cell totally collapse or maybe dead. So a little is known of the host protective mechanism in amoebiasis. There are serum antibodies in this patient. So these serologic antibodies can be used to diagnose amoebiasis. Their role in protection is not clear. Trophozoites are known to activate the complement system via both alternative and classical pathways. Complement is amoebicidal, but the pathogenic strains are resistant to complement medicated lysis. So these are the technical terms used for diagnosis or for detection of the Entamoeba histolytica. So with this, I conclude my lecture, and this is the bibliography I use for preparations of this lecture. Thank you.